So I have a question regarding sample sequencing method.
Okay.
You have mentioned that we use DNTPs for DNTPs along with the DDNTP. We have four reactions, and for an A reaction we use DDATP. That's it, sir. And how would if the sequence has more A's in it, if the reaction terminates at first A, then only that band will be, I mean, like it will show up in the final sequencing. How would we get different bands with different lengths if the reaction terminates at the first A itself? If we do not have DATP in the reaction.
So there is a particular composition of A. You don't add the, for example, you don't add the 25% of A, 25% of G, 25% of C, and 25% of C. You don't add them in an equal proportion actually. You add the A at a very, very low quantity, especially the DATP. So you add all of them at 25%, 25%, 25%, but you add this DDATP at a very low concentration. So because you add a very low concentration, sometimes the enzyme is not going to incorporate, or sometimes the enzyme will actually go through because you are adding the DDATP at a very low concentration. So it will actually overwhelm that and it will reach to the second point. So that's why going to, when you do the PCR, right, with the enzyme, probability wise, sometimes it will stop at first nucleotides, sometimes it will stop at 4th nucleotide, sometimes it will stop at 10th nucleotide, something like that. But it will stop at every A wherever it is present. So it will only going to present that into the gel. Even if it stops 10 times at A, it is not going to give you separate band. It's only going to give you one band. So even if it runs once to the 4th point, that will count into a number of bands. So at some probability wise, sometime it will reach to 4, sometime it will reach to 10, sometime it will reach to 20, like that. And remember that with the single sequencing method, you cannot sequence a very large DNA. You can only do very small fragments sequencing. Is it clear? Clear? Okay, Sarvesh?
Sir, I just want to know about the ongoing studies regarding CAR T cell therapy and about monoclonal antibodies by recombinant technology, sir. Like, what's the government doing?
CAR T cell therapy and monoclonal antibody production is related to the course what we are discussing right now?
Like, where we are using actually retroviruses for generating CAR T cell. So I just want to know what type of viruses they're using, or is there any government work going on?
I have no idea actually. CAR T cell.
Okay, sir. Okay. Thank you so much.
Any more questions?
Hello.
Deepak?
Hello, good afternoon, sir. Sir, I want to ask, sir, what type of insect generally we take as a host?
Can you repeat again one more? What?
So what type of host we take as an insect host for the transfection? So while we are using retroviruses, we have to take insects. So what type of insect we generally take for cloning purposes?
Mm, uh, can you repeat? Actually, voice is very low actually.
Okay, okay. Sir, uh, for cloning purposes, you said we use baculoviruses to insert our DNA into the insect host. So, so whether we are using cell lines of the insect or any special type of insect we are using for these purposes?
No, we are not using the insects. We are using the insect cell line. We are using the actually the cells. So these
Okay,
are SF9 cells.
okay. Thank you, sir. So one
SF9
more
cells.
question. Okay, sir. So thank you, sir. So from yesterday's class, so how would we screen the genomic library that the library is representing each and every segment of the genome? How we will get this information, sir?
You will screen only if you suppose if you get a DNA fragment, right? Which you think, and you are not getting a complete gene. You are only getting a fragment of the DNA, which you are finding whenever there is a that particular type of phenotype is found. Okay. For example, if somebody is developing a diabetic-like conditions, and when you do the subtractive analysis, you found that okay, there is a DNA fragment which is always been present, right? So then what you can do is take those DNA fragment and prepare the probe using that DNA fragment, either by adding the radioactivity or something, and then use that to screen the to complete genomic library to fish out which gene it is belonging to. Because you don't know the genomic sequences, that's why you
Yes.
cannot use the genomic sequences information.
Okay. Okay.
So genomic
Thank you.
library and the cDNA library was more relevant when the people were not having the information about the genomic sequences. But now if you suppose I fish out one particular DNA and suppose I have the information about genetic sequence, I just sequence that gene and blast. So that's how I can get the complete genome sequence.
Okay. Thank you very much. Thank you very much.
Any more questions? Okay, so it's already 4:30 now, 4:20. So if you don't have any more questions, okay, I will going to wind up the session. Uh, Remember that this is an online workshop, so online workshop always have limitations that it cannot give you the real feeling of how to do these experiments. But very soon in the summer, probably we are going to start an offline course, an offline workshop about the same content, or maybe basically the more advanced because that will be a 1 week workshop, and that time probably you will have a better experience of learning all these things. So if you don't have any more questions, I will wind up the session now. And thanks for joining the workshop. And I'm very happy that very enthusiastically you have participated during the course of this whole discussion. And I hope you will could have been some learning experience. And with this, I think I would like to wind up the session now. Okay, thank you. Now you can log out actually.
Hello.
Simple, you want to ask something?
Yeah, sir. Hello, sir. Hi.
Yes,
I
sir.
want
Ask.
to
I wanted to ask you that I have seen that this course of yours on molecular cloning, this molecular biology is available on NPTEL, and we can join this or we can enroll in this course. I wanted to ask you about the exam. Is it going to be held in India or we can have it online, the examination of this course?
You're asking about the MOOC course?
Yeah, yes, sir.
MOOC course is going to be offline. You might have to go to exam center where you might have to attempt the questions on a computer, and so it will be offline in the center which is closer to your city.
Okay, okay.
So it will
Because
not be online.
the exam cannot be held online.
Yes, exam cannot be held online.
Okay, thank you so much.
Yeah. Okay. Bye bye.
Bye.